Okay, so today I'm going to talk about Genomics to Proteins Portal, which is a platform for connecting genetic readouts to protein sequence and structure. So, great. We, we live at an incredible time in terms of the sheer volume of biological data that we're getting. On protein structure, we have advances in cryo-EM that are giving us ever-growing high-resolution complexes. And on the AI side, breakthroughs in deep learning have given us an incredible number of predicted protein structures. Recently, we've also had base editing technologies, which have allowed us to conduct high-throughput functional screens and give us large numbers of data points on the functions of mutations. And on the genomic side, advances in sequence technology have resulted in an exponential growth in sequencing data. And here we have a plot. We saw a previous plot also on this fast growing number of variants of uncertain significance. So the question that we're faced with is, how can we effectively integrate all of this data and put it to use? And I think from the AVE Alliance perspective and from some previous talks, we've seen very well how we can connect all of this mutational scanning data and bring it to protein sequence and then to protein structure. So recently in a base editing study from Brian Lau's group, the group identified essential sites by doing a base editing screen and mapped them to protein structure. So given the value of connecting the genetic variants to protein structure and function, what's stopping us from doing it? What does it take to connect structure and function? As it turns out, it's not that easy. First, you have to get access to the relevant data sets that you're studying for your analysis. You have to navigate through different portals, find the data, download different spreadsheets, write scripts, manually transfer data, compile new spreadsheets. Now you can write a workflow that will take your spreadsheets and visualize them on the sequence. You'll probably use Python or R or Excel. And then finally, you can go to your structure viewer of choice, for example, PyMol, and view the protein structure. The process is pretty time-consuming. We often find that it takes somewhere from a few days to a few weeks to get all of the data together, process it, and get it into the correct tools for mapping. So to address this, we need a tool that will integrate all of these data sources and allow us to easily map our variant data to protein sequence and structure. And to do this, we developed Genomics to Proteins Portal. Genomics to Proteins Portal is a platform that integrates gene, transcript, protein isoform, and protein structure data. And just as a quick example, we can see the ADAT gene here, and we see that it has 4 transcripts, a large number of crystal structures, 2 protein isoforms, and this new AlphaFold predicted structure. So Genomics to Proteins Portal is a resource that brings together researchers from across fields. It gives structural biologists an easy platform to work with genetic screening readouts, and it gives clinical geneticists an easy way to work with protein sequences and structures and map their data over. So how did we construct the portal? So we went through a large number of databases, including genomic, proteomic, transcriptomic, and protein structure databases. Just by including AlphaFold, we saw a substantial increase in our ability to map variants over to structure. From AlphaFold alone, I don't know if it's blocked, but we mapped over 7 million additional variants to residues that didn't have experimental structures. And now let's go through how we could actually use the portal. So the portal has 2 core modules. And the first of those is this gene protein lookup module. And you could search through any of the over 20,000 genes in the human proteome. And the portal will internally map between the gene, the sequence, the transcript, protein isoform, and protein structure. So you can map your variants directly to protein structure. And so let's look at an example here. So CBS, or cystathionine beta synthase, is an enzyme which catalyzes the reaction to convert homocysteine to cystathionine. and rare variants in CBS are the primary cause of homocystinuria, and which is a rare disorder where the body can't properly metabolize methionine. So here we search the CBS gene, and then we're going to look at the sequence features. And so we could see there's some MAVE readouts down here. And you could see going from about residue 100 to about residue 400. You could see that there are this loss of function in the MAVE screen. And then we add additional annotations to the sequence. So here we added physicochemical properties, uniprot annotations, and post-translational modifications. And from the PTMs at the bottom, we could see that the ubiquitination sites seem to line up. So there's a potential correlation there. And I'll say G2P is a hypothesis generating tool. So when you see that, then you're going to go deeper with it and look at it. It doesn't, yeah. So now we've looked at this on the sequence. We have here's additional data, and you could link to MAVEDB and see a lot of additional data on the specific MAVE if you want to go further with it. But now we want to map it to structure. So it's easy to do. You just click on the track in the site, and it maps over to the structure. And here we map the average scores for each of the amino acid positions in the MAVE over onto the structure. And we see some clear clustering. So on this is a dimer structure, and on the dimer, we see that all of the loss of cell growth scores are in this dimerized domain. So, okay. So we've seen how to use the portal to investigate existing databases. But what do you do if you have your own data and you want to use the power of the portal to investigate that? So for that, we developed this interactive mapping module. And it allows you to concurrently analyze your own data alongside the existing databases. And it has a simple workflow. So when you go in, you select a structure, or you could upload your own structure. And then you can upload your annotation data. It's secure. It's all local to your computer. It doesn't go to our servers. I can demonstrate to you how you could verify that. And then you could go and do your analysis. So we're going to go through an example with TRP12. And so TRP12 is a ubiquitin ligase. It works in DNA damage response, and it has variants associated with multiple different diseases. So first, we'll select a structure. You could select an existing PDB structure, an AlphaFold structure, or you could upload any other structure. Then you upload annotations. You can upload annotations from different spreadsheet formats, and the portal will detect different variant formats, scores, or multi-class annotations. And then finally, you can visualize. So for TRP12, we concurrently mapped a novel set of missense variants that were identified in a recent paper on Clark-Beratzer syndrome. And then we mapped them alongside HGMD database variants and ubiquitin sites. And we found that both the new variants and the existing HGMD variants clustered on, this is the AlphaFold structure, and both sets of variants were co-located with ubiquitin sites. It's hard to see that the variants are right next to ubiquitin sites. They are. So ultimately, the interactive mapping tool is a flexible and powerful tool for hypothesis generation. So to circle back and recap a bit, we've seen how the portal integrates functional and structural data across data sources. It provides an easy integration, both as a data resource and as an interactive module. In addition, there are robust and scalable APIs. So you can develop computational methods on top of the mappings that we've put together, and you could scale them across the entire human proteome. And we have a rigorous and routine update protocol. So the portal will stay up to date. We track and take care of frequent updates across all of the large databases, and we track a variety of different update schedules that they have. So right now, we have somewhere over 2,000 users, and We're working as hard as possible to address any and all issues, so please give us any feedback or any issues, challenges that you're facing, and we're happy to work with you. And while the portal has a lot of great functionalities, we have some more capabilities that we're working on to expand the usability. So first, we're working to add mapping across species. So you can conduct your screening on a different species data set and then map it over to human proteins. We're adding tool integrations. So you could do structure prediction, pocket prediction, or mutational impact prediction from the portal, and then view the data on the structure. And finally, we're always adding new data sources. So right now, we're currently working on adding protein-protein interaction sites and pocket annotations. So with that, I have a few thank yous. So first and foremost, I'm really grateful for my PI, Sayeh Iqbal, and my inspiring computational biology teammate, Silky, on the BCB team. And I want to thank the Mutational Scanning Symposium organizers for making this all possible. And finally, I want to thank our funders, especially the Merkin Institute for Transformative Technologies. And one last thing is that I'm really excited to announce that our paper on Genomics to Proteins Portal was just accepted to Nature Methods. So stay tuned, and it will come up shortly.
That's really great. Yeah, from bitter experience in my own life, building those tools, not nearly as good as that over the years. So I know how painful it is. So many, many thanks for that. It looks amazing. Questions? Well, you can find each other and go and demand new things from him. He'll immediately implement at the poster session. Or did somebody put their hand up? No. Should we just say thank you and move on? Yeah. I mean, I think that one thing, I think that I think the whole community will be really grateful, especially the interactive and being able to upload your own things. I think one useful thing, we've been thinking about this is across many groups, is when you're doing the AlphaFold is to also put the nearest PDB up. Right. Because it's sometimes, one amino acid different, and there's a lot more information in them. Okay. Right,
agreed. There are a lot of existing PDB structures. For a lot of the complexes, there's a great cryo-EM complex. So you're looking at an AlphaFold structure, but you could just switch over to the cryo-EM in that case. It's good to have basically you have your options. Yeah, thank you. Okay, thank you very much.